Hello, it's Shudena Nagar, and today we're going to be solving May-June 2023 Paper 1 Variant 2. And before we start, I'd highly appreciate it if you would take a moment to subscribe to this channel and like this video. We have some incredible news to share with you. We are thrilled to announce the launch of our brand new website. And guess what? It comes with an exciting bonus. Free notes for topics 1, 2, 3, and 4. Simply log in and press File Share to receive your free notes. And the link to the website would be in the description box down below. Question number 1. A graticule and a micrometer scale can be used to measure the size of biological structures that are viewed with a microscope. Which row shows the correct locations for the placement of a graticule and a micrometer scale on the microscope shown? So let's start with a graticule. The full name of a graticule is eyepiece. Graticule. Therefore, it is placed on the eyepiece. What it is, it's basically a glass slide with no calibrations and without units. However, it has equal distance and equal measurements between each other. However, it is not calibrated and it's without units. Contrasting a stage micrometer, which from its name is placed on the stage, which is number 3. Its function is that it has known dimensions and units. Therefore, these could be measured to calculate the magnification of an object. Therefore, the answer is going to be B. Question number 2. 6 organelles found in eukaryotic cells are shown. Which organelles are involved in the synthesis and secretion of a glycoprotein? So let's outline the process of protein synthesis. It starts in the nucleus, in the process called transcription, where mRNA is formed. And then this mRNA leaves the nucleus into the cytoplasm and goes to the rough endoplasmic reticulum, in particular on the ribosomes, where this mRNA is translated into a polypeptide chain. Now, what happens is that we've now formed a polypeptide, and now we want to form a glycoprotein. Therefore, what happens is the vesicles on the rough endoplasmic reticulum bud off and go to the Golgi body, where the Golgi body adds the sugar part and creates the glycoprotein. And by the way, the function of Golgi body is to modify proteins. Therefore, the organelles that we need are mainly the nucleus, the rough endoplasmic reticulum, in particular the ribosomes on the rough endoplasmic reticulum, and Golgi body. However, in order to do this process of protein synthesis, we need an energy source, aka ATP. And now, what forms that energy source is the mitochondria. Therefore, now we have the nucleus, the mitochondria, rough endoplasmic reticulum, and Golgi body. And the reason why this is the Golgi body, not the rough endoplasmic reticulum, it is that it's a layer of flattened stacks. Therefore, this is the Golgi body, whilst the rough endoplasmic reticulum has ribosomes. Therefore, that's how to distinguish them. Therefore, the answer is going to be B. Question number 3. Which cell structures can have mRNA inside them? So let's start with chloroplasts. We know that chloroplasts are found in plant cells and they have 70S ribosomes. And chloroplasts are able to synthesize their own proteins. Therefore, if they have 70S ribosomes, they must have mRNA to be able to translate this mRNA into proteins. Therefore, chloroplasts do have mRNA inside them. Now, mitochondria also have 70S ribosomes. Therefore, they are also able to synthesize their own proteins. However, they require mRNA in order to do so. Therefore, mitochondria can have mRNA inside them. Now, for 3, nucleus. We know that the process of mRNA transcription occurs in the nucleus. Therefore, nucleus does have the mRNA inside of it. Now, 4, rough endoplasmic reticulum. We know that ribosomes or ATS ribosomes are found on the rough endoplasmic reticulum. Therefore, the mRNA is translated on top of the rough endoplasmic reticulum inside the ribosomes. Therefore, rough endoplasmic reticulum do not actually have the mRNA inside them. they have the mRNA inside the ribosomes, which are found on top of the rough endoplasmic reticulum. Therefore, this is incorrect, and the answer is going to be B. Question number 4. A scientist carried out an experiment to separate organelles in an animal cell by mass. The scientist mixed the cells with a buffer solution which had the same water potential as the cells. The cells were broken open with a blender to release the organelles. The extracted mixture was filtered and then spun in a centrifuge at a high speed to separate the heaviest type of organelle. These sank to the bottom, forming a solid pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun in the centrifuge at a higher speed to separate the next heaviest type of organelle. These organelles sank to the bottom, forming solid pellet 2. This procedure was repeated twice more to obtain pellet 3 and 4, each containing a single type of organelle. What is the main function of the type of organelle extracted in pellet 2? So, the theory behind this question, or how to actually answer this question, is we must know the size of organelles. Now, here it says formed pellet 1. We must know that pellet 1, because it separates the heaviest type of organelle, therefore pellet 1 is going to be the heaviest organelle. And this table is from our free topic 1 notes in the website. Now, in this table, we note that the heaviest type of organelle must be the nucleus, since it's the largest. Now, the second type of organelle, or the second heaviest, which is pellet 2, in this case, it's an animal cell, it's going to be the mitochondria. This is the second heaviest in an animal cell. Therefore, pellet 2 is going to be the mitochondria. Now, the main function of pellet 2 is synthesis of ATP, therefore the answer is going to be B. Question number 5. Which structures are found in palisade mesophyll cells and photosynthetic prokaryotes? So let's start with number 1, cell surface membrane. Yes, both palisade mesophyll cells, which are plant cells, and prokaryotes do have a cell surface membrane. Now, for 2, cellulose wall. Now, cellulose wall are only found in plant cells, in this case, and only in the palisade mesophyll. However, in prokaryotes, the cell wall is a peptidoglycan cell wall. Therefore, this is incorrect. 3. Ribosomes. Palisade mesophyll cells have 80S ribosomes in the cytoplasm and 70S ribosomes in the chloroplast and mitochondria, contrasting the prokaryotes, which have 70S ribosomes inside the cytoplasm. Therefore, this is correct. For 4, chloroplasts. Palisade mesophyll cells do have chloroplasts since they carry out photosynthesis, and they are the main cells which actually carry out photosynthesis in a plant. However, even though this is a photosynthetic prokaryote, we all know that in a general rule, prokaryotes do not have membrane-bound organelles. Therefore, it's impossible to have chloroplasts. Therefore, the answer is going to be C. Question number 6. Which polymers are present in all viruses and all prokaryotes? Now, let's start with polynucleotides. So, polynucleotides, the definition of it, are nucleotide polymers joined by covalent bonds. So, whether the virus is an RNA virus or a DNA virus, it must have a polynucleotide. Same with prokaryotes. All these must have some sort of genetic material, and polynucleotide is a general term for any sort of genetic material, whether it's RNA or DNA, because as we said, they are nucleotides joined together by covalent bonds. Now for 2, polypeptides. So let's start with viruses. We know that all viruses must possess a protein capsid. Therefore, all viruses must contain polypeptides. Same with prokaryotes. Prokaryotes have 70S ribosomes which synthesize proteins. Therefore, proteins are present in both. 3. Polysaccharides. Now, not all viruses have polysaccharides. Therefore, this is incorrect, and the answer is going to be B. Number 7. Which set of steps is the best method for conducting the emulsion test for lipids? So, this experiment here is found at the free topic 2 notes, biological molecules, which are also found on our website. Now, the experiment is the ethanol emulsion test for lipids. Now, the procedure is shake and mix the sample with ethanol Why do we do that? Because this allows lipids to dissolve, since lipids are soluble in ethanol. And then we add water to the sample. And now the reason why we add water to the sample is because lipids cannot remain dissolved in ethanol. So they form droplets as a cloudy emulsion, and that's where the theory behind the cloudy emulsion test comes from. Now step 4, if the lipids are present, the solution turns cloudy, and if no lipids are present, the solution remains clear. Therefore, the answer is going to be D, since we are adding ethanol and then pouring the ethanol into water to create the droplets, and then if lipids are present, the mixture becomes cloudy. A student was provided with a solution of carbohydrate. They removed 2 samples from the solution and performed tests on each sample as shown. Which statement explains the result? Now, let's take a look at this experiment. Now, carbohydrate solution, sample 1 and sample 2. Sample 1, they added Benedict's test. Now, Benedict's test is used to test for reducing sugars, and a positive test for reducing sugar would be a brick red color. However, in this case, this solution remained blue. Therefore, there was not any reducing sugar in this sample, so no reducing sugars. Now, sample 2 was boiled with dilute hydrochloric acid, and what dilute hydrochloric acid does is simply break the glycosidic bond. Of a non-reducing sugar. In this case, for example, a sucrose. It breaks the glycosidic bond to turn it into a reducing sugar. Now, neutralize with dilute sodium hydroxide, and then we added Benedict's test, and the solution turned yellow. If we boiled with dilute hydrochloric acid, then the solution turned yellow. This confirms the presence of non-reducing sugars. Therefore, let's see the suggestions that we have here. Condensation reaction occur in sample 2 to release reducing sugar. Now, this is a hydrolysis reaction. Hydrochloric acid breaks the glycosidic bond by hydrolysis by adding H2O. Therefore, this is incorrect. B. Glycosidic bonds in a polysaccharide have been broken to release the reducing sugar. Yes, I've just mentioned this point a few times. C. Sample 1 shows that sucrose is present in the carbohydrate solution. This is incorrect since sucrose is a non-reducing sugar. And this test confirms the presence of a reducing sugar. Therefore, this is incorrect. D. The change in color to a yellow solution shows that glucose is present. We cannot always confirm that it is glucose. The reason for this is there are other types of reducing sugars, not only glucose. Therefore, the answer is going to be B. Question number 9. Which molecules contain at least 2 double bonds? So let's start with sucrose. We know that sucrose is formed by the condensation of fructose and glucose, forming a glycosidic bond. Therefore, they do not contain any double bonds whatsoever. Now, with collagen and hemoglobin, we know that collagen is formed from 3 polypeptide chains, whilst hemoglobin, the heme part, has a polypeptide chain. Therefore, they all have protein components. And as we all know, a peptide bond looks something like that. Therefore, the peptide bond definitely has a double bond. And we know that these are polypeptides. Therefore, they must have many peptide bonds. Therefore, they contain at least 2 double bonds. Therefore, collagen and hemoglobin are correct, and the answer is going to be B. Question number 10. What describes cellulose? So, this part here is found at the topic 2 notes on the website. Now, cellulose definition is a polysaccharide made from beta-glucose subunits rotated 180 degrees to each other, forming a beta 1-4 glycosidic bonds. And it is an unbranched chain since they're rotated 180 degrees. Therefore, let's see the suggestions that we have here. A. A branched chain of 1-4 alpha glucose. Now, this is incorrect. It is beta glucose, and at the same time, it's not branched. It's linear. Now, this is incorrect since it's alpha glucose, and therefore, D is going to be the correct answer. Question number 11. Which part of the structure of hemoglobin carries oxygen? So we know that hemoglobin is made up of 4 polypeptide chains, each containing a heme group. That heme group contains iron, which indeed binds an oxygen molecule. Therefore, a hemoglobin made up of 4 polypeptide chains containing 4 heme groups would carry in total 4 oxygen molecules. Therefore, the answer is going to be B. Question number 12. What is the maximum number of hydrogen bonds that can form between two single water molecules? Now, I'll try explaining this without getting into heavy chemistry. Now, let's draw a water molecule. We know that oxygen has a total of 6 electrons and hydrogen has 1. So let's draw them out. Therefore, for oxygen, we have a total of 4 electrons left, which are drawn in pairs. Now, these 4 electrons are able to also form hydrogen bonds with other water molecules. And these hydrogen ions are also able to form hydrogen bonds with other water molecules, but with the oxygen part. However, here the question states, what is the maximum number of hydrogen bonds that can form between two single water molecules? Therefore, let's assume that these are two single water molecules. The answer is going to be 1. However, water can form hydrogen bonds between 4 water molecules. Therefore, they can form 4 hydrogen bonds. However, because the question is asking between two single water molecules, it's going to be 1. Therefore, the answer is going to be A. Cytochrome 3A4 is an important enzyme in the human digestive system where it is needed to break down a range of different toxins. So the substrate for this enzyme are the toxins. Now, the activity of cytochrome 3A4 has been shown to be reduced by the substances called furanocoumarins. Furanocoumarins are found in some fruits, and so dangerous concentrations of toxins may develop in the human digestive system when fruits containing furanocoumarins are eaten. So, furanocoumarins are potential inhibitors for that enzyme. However, the question is not specifying whether this is a competitive or a non-competitive inhibitor, since it did not mention whether increasing the concentration of the toxin overcomes the enzyme inhibition or not. Now, from the information provided, what can be concluded about molecules of the enzyme cytochrome 3A4? Now, let's see the suggestions that we have here. They lower the activation energy of the toxin breakdown reactions. So we know that features of any enzyme is that it lowers down the activation energy, or the energy needed to start a reaction, by providing an alternative pathway with a lower activation energy. Therefore, this is correct. For B, they bind specifically through the active site to a substrate found in some fruits. So we've just mentioned that the substrate for the enzyme are the toxins, not the fruit. The furanocoumarins are inhibitors, like furanocoumarins found in fruits are inhibitors to that enzyme. They are not these substrates. Therefore, this is incorrect. And also, binding specifically through the active site, I've just mentioned that it is not specified whether this is a competitive or non-competitive inhibitor, so in both ways this cannot be correct. For C, they change permanently when acted upon by furanocoumarin molecules. We just mentioned it is not specified whether it's competitive or non-competitive, therefore this is incorrect. For D, they resume normal activity when concentrations of furanocoumarins decrease. This would be correct if this were a competitive inhibitor. However, because it's not specified, this is incorrect, and the answer is going to be A. Question number 14. A fixed volume and concentration of substrate and enzyme were mixed. All other variables were kept constant. The enzyme catalyzed reaction was left until it was complete. which graph shows how the rate of reaction changes with time. So as we all know, at the start of an enzyme-substrate reaction, what happens is that the rate of reaction is the greatest. The reason for this is that the substrate concentration is high, therefore all the active sites or most the active sites of enzymes are saturated, therefore the rate of the reaction is the highest at the start. And now as time goes on, the substrate concentration decreases. Therefore, the rate of substrate enzyme collisions decrease. Therefore, this keeps lowering the rate of the reaction over time. Therefore, a more suitable graph would be C. Since the rate of the reaction, you must carefully look at the axis. The rate of the reaction was the greatest at the start, and as time goes on, it kept decreasing due to the reaction which I explained. Therefore, the reaction is going to be C. Number 15. Which molecules in cell surface membranes are typically involved in cell recognition? So, cholesterol. Cholesterol is involved in regulating the fluidity of the membrane, not really involved in cell recognition, therefore this is incorrect. Now, glycolipids and glycoproteins. So, glycolipids and glycoproteins are found outside the cell membrane and they are involved in cell recognition, for example, as receptors. Therefore, this is correct, as receptors and antigens. Now, for phospholipids, the function of phospholipid is to form the cell surface membrane or the lipid bilayer, and the function of phospholipids are to prevent the entry of hydrophilic substances. Therefore, the answer is going to be C. Question number 16. What can increase the fluidity of the cell surface membrane? 1. Single bonds between carbon atoms in the fatty acid chains. So as we all know, unsaturated fatty acids or double bonds actually increase the fluidity of cell surface membrane, whilst the single bonds decrease the fluidity. Therefore, this is incorrect. 2. Cholesterol. So when the temperature is low, cholesterol actually increases the fluidity of cell surface membrane. And when the temperature is high, cholesterol lowers the fluidity of cell surface membrane, and it acts as a fluidity regulator inside the cell. Therefore, at low temperatures, it actually can increase the fluidity of the cell surface membrane. Therefore, this is correct. For 3, longer chained fatty acids. Now, this is incorrect. The reason for this is longer chain fatty acids means that there are going to be more bond interactions, and the more bond interactions actually decrease the fluidity since there are stronger bond interactions. Therefore, this is actually incorrect. It decreases the fluidity instead of increasing the fluidity. Therefore, the only correct answer is going to be D. 17. The 3 main factors that affect the rate of diffusion across a membrane can be expressed by the relationship shown. Which changes in the factors would result in the rate of diffusion doubling? So, we know that the rate of diffusion is inversely proportional to the thickness. So, if the thickness decreases, the rate of diffusion increases. And the surface area and concentration are directly proportional to diffusion. Therefore, if they increase, diffusion also increases. Therefore, let's see the suggestions that we have here. Now, surface area has doubled. So here we're looking for the factors that result in the rate of diffusion doubling, so increasing. Now, surface area has doubled. As we said, they are directly proportional, so this is correct. 2. Concentration difference has halved. So, if the concentration difference has halved, because they are directly proportional, also the rate of diffusion would halve. Therefore, this is incorrect. 3. Thickness of membrane has doubled. Now, if the thickness of membrane has doubled, then the rate of diffusion would halve because they are inversely proportional. Therefore, this is incorrect. 4. Thickness of membrane is halved. So, the thickness is halved, therefore, the rate of diffusion would double because they are inversely proportional. Therefore, 1 and 4 is correct and the answer is going to be C. Question number 18. A student measured the time taken for complete diffusion of a dye into agar blocks of different sizes which were suspended in the dye. The results are shown. What is the predicted time for complete diffusion of the dye into the agar block measuring 5 to 10 to 15 millimeters? So here we're looking for an estimate. We're not looking for the specific time. Now, that estimate will become when we know the surface area to volume ratio, which is the closest to the surface area to volume ratio of this block, or block 4. Therefore, in order to actually predict the time and answer the question, we must calculate the surface area to volume ratio for each of the blocks. So let's start with the first. Now, for the first block, the surface area in this case is going to be 150, and the volume is going to be 125, giving us a surface area to volume ratio of 1.2 to 1. Now for block 2, the surface area is going to be 600, and the volume is going to be 1000, giving us the surface area to volume ratio of 1 to 1.6. Now for 3, the surface area is 1350, and the volume is 3375, which gives us a surface area to volume ratio of 1 to 2.5. Now for block 4, the surface area is going to be 550, and the volume is going to be 750, giving us a surface area to volume ratio of 1 to 1.3. Now, we have to look for the surface area to volume ratio which is closest to 1 to 1.3. Therefore, making 1 the most suitable, since 2 is 1 to 1.6, which is way further, and 3 is 1 to 2.5. Therefore, the time would be too long. Therefore, we are predicting that the time would be quite similar to 1. Therefore, the answer in this case would be 6.2, which is A. 19. An experiment was carried out to investigate the effect of concentration of sucrose solution on cells in a plant tissue. A sample of a plant tissue was cut into 7 cylinders of equal length and diameter. The mass of each cylinder was recorded. Each of the 7 cylinders was put into different sucrose solution concentration. After 2 hours, the cylinders were removed, blotted dry, and reweighed. The percentage change in mass of each cylinder was recorded. The graph shows the results of this investigation, which explains the results if plant tissue cells were put into sucrose solution of 0.5 mole per decimeter cubed. Now, 4.5 mole per decimeter cubed is somehow here. And this shows that the percentage change of a plant is with the negative. Therefore, water moved out of the plant. Water moved out of the plant because the water potential outside were less. Therefore, the water potential inside the plant at that concentration was higher for water to move out. Therefore, the water potential of the cytoplasm of these cells at the start of the experiment compared with the water potential of 0.5 mole decimeter cubed sucrose solution is definitely going to be less negative. Less negative means that it has a higher water potential. So a less negative potential is a higher water potential, and we just mentioned here why. Now, the change in volume of vacuoles of the cells at the end of the experiment that were initially placed in 0.5 mole decimeter cubed sucrose solution. So, the change in volumes is going to be decreased since the water left these cells. Therefore, the answer is going to be A. Question number 20. The diagram shows part of the organization of a section of a DNA molecule and the associated histones P and R in a prophase of mitosis, with statements about the features labeled P, Q, and R during prophase of mitosis are correct. So let's identify Q. Q is the DNA molecule. Since the DNA is incredibly long, it must be wrapped around proteins called histones to be compact to fit inside a very tiny nucleus. Now let's see the suggestions that we have here for A. The cold DNA molecule forms Q, So this is correct, and wraps around the histone R to form small clusters held in place by histone P. The wording here is incorrect. It wraps around the histone P to form small clusters held by histone R. Therefore, this is incorrect. Plus, this is also incorrect because here they're asking specifically during prophase of mitosis. This happens all the time, not just during prophase. Therefore, for those two reasons, this is incorrect. B. The groups of histones P and its associated DNA, Q, move closer together as the chromosome condenses around R. This is totally incorrect. The reason for this is, first of all, the histones and the DNA wrapped around the histones forms something called a chromatin with an N. These three components form a material called chromatin. Therefore, a chromatin, or this long strand of DNA and histones, form the chromatid, which is one part of a chromosome. So a chromatid. So the chromatin, or this material, wraps around to form a chromatid. So what happens is that the chromatin, or the super long strand, is what actually condenses together, not the histones that come closer together. There's a huge difference. Therefore, this is incorrect. D. The linked groups of histones P and R and the associated DNA Q form strands that fold and twist together to form a chromatid. Yes, this is totally incorrect. I've just mentioned that this material is called the chromatin, and chromatin is the material that forms a chromatid, which forms the chromosome. Therefore, it's the chromatid that wraps around each other, not the histones that condense together. This is incorrect. The histones and the DNA stay in shape and they do not condense or move. Therefore, this is correct, and the answer is going to be D. Number 21. How many copies of each DNA molecule will be found in a cell at the start of the stages of mitotic cell cycle shown? So, the cell cycle is made of mitosis, which is made of prophase, metaphase, anaphase, and telophase. Then after this is something called cytokinesis, which is the division of the cell. Now, here, this is called interphase. And the interphase is divided into three parts, which is G1, S phase, and G2. In G1, the cell continues to grow and protein synthesis takes place. In the S phase is actually where DNA replication takes place. So after the S phase and before the cytokinesis, when the cell divides during this area here, is where you would find two copies of the DNA molecule. In this area here, the area after DNA replication, which is the S phase, and area before the G1. Therefore, let's see the suggestions that we have here. Now, for G1 of interphase, how many copies we would have? We just mentioned that G1 is before the S phase and it's after the cytokinesis. Therefore, it's only going to be one copy. Now, for cytokinesis, at the start of cytokinesis, the cell is still preparing to divide, therefore it's going to still have the two copies. However, at the end of cytokinesis, when the two cells have actually divided and the genetic material was divided, it would have only one DNA molecule or one copy. Therefore, the answer is going to be B. Number 22. One characteristic of DNA is that it is a universal genetic code. What is meant by a universal genetic code? So, it is a genetic code made of codons. So codons are 3 nucleotides, and each codon codes for one amino acid. So let's see the suggestions that we have here. A. All living organisms use the same triplet code for amino acids. Yes, this is correct because we've just mentioned that the genetic code codes for codons that makes universal amino acids. Therefore, this is correct. For B, all DNA triplets code for a different amino acid. Now, this is incorrect. This defeats the meaning of a universal genetic code, which all codons codes for the same amino acids. C. Not all DNA triplets code for an amino acid. This is incorrect. D. All living organisms contain the same 4-nucleic acid. So, cytosine, guanine, adenine, and thymine. This is totally incorrect because for viruses, as an example, we have RNA viruses. RNA viruses have uracil instead of thymine. Therefore, not all living organisms contain the same 4-nucleic acids. Therefore, this is incorrect, and the answer is going to be A. Number 23, which statement about mRNA is correct? A. It is a polymer made of nucleotides all joined with hydrogen bonds. It is incorrect. Nucleotides are joined together by phosphodiester bonds, not hydrogen bonds, which makes them very strong since they are covalent bonds. Now, for B, each nucleotide subunit contains the sugar ribose. This is correct. RNA contains ribose sugar, whilst DNA contains the oxyribose. Therefore, this is correct. C, it always has an equal proportion of adenine and uracil. This is totally incorrect. We're not here talking about something that is complementary. Here we're talking about a single strand. Therefore, it could have any proportion. It doesn't have to be equal. Therefore, this is incorrect. D, the mRNA sequence is identical to the template strand of DNA. This is also incorrect. They are complementary, not completely identical. So, if mRNA has adenine, DNA would bind with a thymine nucleotide because they are complementary. Therefore, this is incorrect, and the answer is going to be B. Number 24, the diagram shows part of the process of translation. What are the names of the structures labeled X, Y, and Z? So, first of all, you must identify that this is a tRNA, and this is an mRNA. An mRNA, 3 nucleotides are codons, and this part is an anticodon. Now, the tRNA carries an amino acid which is complementary to the codon. So this is an amino acid. Now, what are the names of the structures labeled X, Y, and Z? Now, for X, we just mentioned that this is an anticodon, which is complementary to the codon. So this is correct. This is an anticodon, which is found in the tRNA. Now, for Y, it is the codon, and for Z, we've just mentioned that it is the tRNA. This whole structure is called the tRNA, therefore B is going to be the correct answer. Question number 25. The DNA triplets of genes are translated as amino acids or stop signals during protein synthesis. The table shows some of these triplets. What could be the effects of one substitution mutation in a triplet coding for tyrosine? So, a substitution mutation is a type of point mutation. So, point mutations are any mutations affecting a single nucleotide. So, in this case, we have substitution mutations, deletion, and insertion mutations. So substitution mutations is where one nucleotide is replaced by another in the codon, and deletion is when a nucleotide is completely removed from a codon or deleted. Insertion mutation is where a nucleotide is inserted into a codon. So let's see the suggestions that we have. 1. The triplet is translated as cysteine. So here we have tyrosine, ATA, ATG, and cysteine as ACA, ACG. So a substitution mutation is when one nucleotide is replaced by another. In this case, tyrosine could turn into cysteine if the thymine is replaced by cytosine. So this is correct. For 2, the triplet is translated as tryptophan. So let's see the suggestions that we have here. So, from tyrosine to tryptophan, this is not possible. Since a substitution mutation, we're talking about only one nucleotide. Therefore, this is not possible. For 3, the triplet is translated as tyrosine. So, from tyrosine to tyrosine, if you're lucky enough, this is actually possible. So, if a substitution mutation happens and adenine turns into guanine, yes, tyrosine could also be translated into tyrosine because apparently in our genetic code, multiple codons could code for the same amino acid. Therefore, this is correct. For 4, translation stops at this triplet. This means that the substitution mutation caused a stop signal. So, this is possible if, for example, the ATA, so the adenine, was substituted with cytosine, then this is possible to turn into a stop signal. Therefore, 1, 3, and 4 is correct, and the answer is going to be C. Question number 26, which row identifies cells with plasmodesmata? So, plasmodesmata, the definition of it, are cytoplasmic channels. So, most live cells with a cytoplasm, and it's a plant cell, is most likely to have a plasmodesmata. So let's see the suggestions that we have here. Phloem sieve tube elements. So phloem sieve tube elements are live cells with a cytoplasm, therefore they do have a plasmodesmata. Now companion cells also have plasmodesmata, which is also needed for the sucrose to leak into the phloem sieve tube elements by diffusion. Now, for xylem vessel elements, we just mentioned that plasmodesmata are cytoplasmic channels, and xylem vessel elements are dead cells without a cytoplasm. Therefore, they do not have plasmodesmata. Plus, if xylem vessel elements were to have plasmodesmata, this would allow resistance in the water. Therefore, water transport will not be as smooth, and water would actually leak out of the vessels. Therefore, xylem vessel elements do not have plasmodesmata for a reason. Therefore, the answer is going to be B. Question number 27. Which statements about the apoplast and symplast pathways are correct? So, the apoplast pathways is the pathway that takes place, water transport, outside the cell walls of the plant. So, the water does not actually enter the cell itself. Whilst the symplastic pathway is the water transport method, or the transport method where water actually enters the cell through the plasmodesmata. Now let's see the suggestions that we have here. 1. In the apoplast pathway, water molecules move through free spaces in the cellulose cell walls of plant roots. So this is correct because I've just mentioned that the apoplastic pathway is the transport of water through the cell walls. Therefore, this is correct. 2. In the symplast pathway, water molecules diffuse through the cytoplasm and plasmodesmata of cells. This is correct because water actually enters inside these cells. For 3, water molecules traveling through the plant tissue move by mass flow along the apoplast pathway. Now this is correct. Water moves by mass flow, and mass flow by meaning large quantities in bulk. Therefore, this is correct. 4. The Casparian strip blocks the symplast pathway. Now, this is totally incorrect. The Casparian strip blocks the apoplastic pathway to force water in the symplastic pathway. Therefore, this is incorrect, and the answer is going to be B. Question number 28. Where does water evaporate from during transpiration? So the answer is going to be mesophyll cells. The reason for this is that the mesophyll cells are the main photosynthetic cells found in the plant leaf. Therefore, they produce the most amount of water since this is the product of photosynthesis. Therefore, water would evaporate from the plasmodesmata into the cell walls of the mesophyll cells. Therefore, the answer is going to be C. Question number 29. The diagram shows the outline of 3 xerophytic leaves of the same type in the 3 different conditions P, R, and S. Which description of the water potential of these cells in layer Y is correct? So in P, we could clearly see it's turgid, therefore the water potential is going to be the highest, whilst in Y or in R, the water potential here is going to be the lowest, and S is in the middle. Now let's see the suggestions that we have here. So, water potential of cells in layer Y in P. So, higher than S and R. So, this is correct. And it could also be higher than S, however, it's higher in both. Now, for R, lower than P and S. This is correct. It is lower than P and S because it is the most curved, indicating that it has the lowest water potential. Therefore, this is correct. And for S, we just mentioned it's in the middle. So it would be lower than P and higher than R. Therefore, the correct answer is going to be A. Question number 30. The photomicrograph shows a transverse section of the leaf of a species of grass. The grass is specially adapted to grow in a dry habitat, which correctly explains how the features help the grass to grow in this habitat. So this is a xerophytic plant, and these structures or hair-like structures are called trichomes. The functions of these trichomes is that they create a layer of still air inside the leaf, therefore reducing wind speed, therefore reducing evaporation, therefore helps with water loss control. Now, what happens is that the internal humidity, because we created a layer of still air inside, increases. And the leaf shape, because it's curved, it also helps with creating a layer of still air to reduce evaporation, therefore also increase the internal humidity, since we're having a layer of still air. Therefore, the answer is going to be C. Number 31, which statement supports the theory of active loading of sucrose into companion cells? So let's quickly state that process. So this is the phloem sieve tube elements, and that's the companion cell. So the companion cell, outside of it are found these sucrose molecules. Now, what the companion cell does is that it pumps hydrogen ions in an active process, therefore reducing the pH at the cell wall, because hydrogen ions, when they increase in concentration, this reduces the pH at the cell wall. These hydrogen ions indeed combines with a sucrose molecule and goes back inside the companion cell via a transport protein. So hydrogen ions going against concentration gradient, while sucrose going down concentration gradient. Therefore, when sucrose is now inside the cell, it moves by simple diffusion inside through the plasmadesmata into the phloem sieve tube elements. So let's see the suggestions that we have here. A, the pH decreases. So this is correct. So in the cell wall of the companion cells compared with the cytoplasm. So this is correct, because hydrogen ions are actually pumped into the cell walls. Therefore, the answer is going to be A. Question number 32. Which properties of water are essential for its role in the transport of blood in mammals? So here we have high latent heat of vaporization and solvent for polar molecules. So latent heat of vaporization, the definition of it is the energy it takes to evaporate a substance. So here, as a role in the transport of blood, we do not need evaporation. It would be correct if it was a higher specific heat of capacity, because a specific heat capacity is the energy it takes to increase the temperature of a substance by 1 degree. So if this were high, this helps maintain a constant temperature inside the body. However, here in high latent heat of vaporization, this is incorrect, because here we're not dealing with blood evaporating. Therefore, this is totally incorrect. Now, solvent for polar molecules. So being a solvent helps it transport substances such as hemoglobin and waste substances. Therefore, this is correct. And the answer is going to be B. Question number 33. The diagram shows some of the events that happened between the plasma and the red blood cells in the circulatory system. What do numbers 1, 2, 3, and 4 present? So, numbers 1, most likely it's going to be the carbon dioxide. Since the carbon dioxide is dissolved in the plasma, then it enters the cell, combines with water to form carbonic acid. This reaction takes place by an enzyme found in the red blood cell called carbonic anhydrase. So, carbonic anhydrase. When carbon dioxide combines with water forming carbonic acid, this carbonic acid dissociates into hydrogen ions, which stay inside the cell, they don't leave, and they dissociate also into bicarbonate ions, which actually leave the cell. So, let's see the suggestions that we have here. And here we do not talk about the phenomenon called a chloride shift. So basically, what a chloride shift is now the bicarbonate, which is negatively charged, is leaving the red blood cell as the concentration of carbon dioxide increase. Therefore, what would happen if negatively charged bicarbonate ions kept leaving the red blood cell? The red blood cell would accumulate an overall positive charge. In order to balance out this phenomenon, there is something called a chloride shift, which is chloride ions found inside the plasma enter inside the red blood cell, so opposite direction to the bicarbonate, in order to balance the charge so the red blood cell does not come positively charged. So let's see the suggestions that we have here. Now for one, we've just said that it only could be carbon dioxide. Now for two, we just mentioned that is bicarbonate, since bicarbonate is the only component that could actually leave the red blood cell. Now for three, we said hydrogen ions because they maintain inside the red blood cell. And for four, due to the chloride shift and it enters the red blood cell, is going to be the chloride ions. Therefore, the answer is going to be B. Question number 34. The diagram shows the valves in the heart when viewed in cross-section from above at different stages in the cardiac cycle. Which stages in the cardiac cycle are shown? So these are the semilunar valves, and they open when the ventricle contracts and close when the ventricle relaxes, or in diastole. The atrioventricular valves open in atrial systole and close in atrial diastole. So let's see the suggestions that we have here. So we just mentioned that the semilunar valve could be closed in ventricular diastole, aka diastole, because when diastole is mentioned by itself, we always indicate the ventricle. Or it could be also closed in atrial systole. Now, regarding the atrioventricular valve, it could be closed during atrial diastole or ventricular systole, or just systole. Now, let's see the suggestions that we have here. So, for A, diagram 1 shows diastole, which is correct. We says that diastole is always referred to, so if it did not specify the diastole, we always refer to the ventricle, so this is correct. And for diagram 2, we said that ventricular systole, the atrioventricular valve, would be closed. So for B, atrial systole, this could also be correct. We said it could be both. Now, for diagram 2, diastole or ventricular diastole. So when it's diastole by itself, we indicate the ventricle. Therefore, this is incorrect because here is ventricular systole. Now for C, this is incorrect. Therefore, the answer is going to be A. Question number 35. Where are the squamous epithelial cells found in the human gas exchange system? So the squamous epithelial cells are only found inside the alveoli, because the other components contain stuff such as stratified columnar ciliated epithelium. So the other organelles in the gas exchange system have columnar cells, not squamous, such as the trachea and bronchus have columnar cells. The alveolus is the only component that actually has squamous cells. Therefore, the answer is going to be D. Question number 36. Which statement about gas exchange between air in the alveoli and blood in the pulmonary capillaries is correct? For A, the oxygen concentration in the capillaries leaving the pulmonary artery is higher than the oxygen concentration in the alveoli. So the pulmonary artery is actually deoxygenated blood, so it will have lower concentration, therefore this is incorrect. For B, gases must diffuse across the endothelium of the pulmonary capillaries and the endothelium of the alveoli. So this is incorrect, the epithelium of the alveoli, therefore this is incorrect. C, the elastic fibers in the alveoli walls allow the alveoli to expand to increase the surface area available for diffusion. This is correct. The elastic fibers are able to expand for the process of diffusion and increasing the surface area, so this is correct. D, breathing out reduces the carbon dioxide concentration gradient between the blood in the pulmonary capillaries and the air in the alveoli. Now, this is totally incorrect. Because the gradient is created by the waste found inside the blood. So the blood has a higher concentration of carbon dioxide since it's waste. Therefore, the gradient stays quite constant. Also, the carbon dioxide gradient found in air is a constant percentage. Therefore, it's least likely to change. Therefore, the answer is going to be C. Question number 37. The average thickness for some structures in the human respiratory system are shown. A molecule of oxygen is in the alveolar airspace next to the wall of the alveolus. What is the shortest distance that the molecule needs to diffuse from its current position to the hemoglobin that completely fills a red blood cell in the nearest capillary, assuming the red blood cell touches the walls of the capillary? So let's draw the pathway out just to make things easier. So the molecule of oxygen is in the alveolar airspace, so it's moving from the alveolar airspace into the capillary. So this is the alveolar airspace. We know that the alveoli are made of squamous epithelial cells. So let's draw out these squamous epithelial cells. Therefore, to start with, we know that if the oxygen molecule is here, it's going to have to move through two human cell membranes. It's going to have to move through two cell membranes just to exit the alveoli. And now here, we have the capillary. We know that the capillary are made of endothelial cells. Endothelial cells have also two membranes. So once the red blood cell passes through the two membranes of the squamous epithelium inside the alveoli, it's going to pass through the two membranes in the endothelium in the capillary. So two here and two here. Now, here is the red blood cell. It's going to pass, in order to enter inside the red blood cell, is going to pass through one cell membrane in order to enter. So here we have one cell membrane. So in total, we have 5 human cell membranes and 1 cytoplasm of alveolar cells, because it needs to just pass through one cell, therefore 1 cytoplasm. And there, it needs 1 cytoplasm of capillary wall cells, because it only needs to pass through 1 endothelial cells, and the tissues between alveolar wall and capillary wall also has to pass through them, which is here. Therefore, what we're going to do is we're going to, first of all, multiply 5 by 5, which is going to be 25, because we just said that it needs to pass through 5 human cell membranes, plus 1 cytoplasmic alveolar wall cells, so 190, and 90, and 300, which is going to give us 650 nanometers. Therefore, the answer is going to be C. Number 38, the statements refer to the disease tuberculosis. Which statements explain why antibiotic treatment for tuberculosis takes a long time? So you must know that tuberculosis replicates intracellularly. This means that antibiotics take quite a while in order to kill all of the bacteria. So let's see the suggestions that we have here. 1, the pathogen is not accessible to the immune system. So this has nothing to do with the antibiotic treatment, therefore this is incorrect. And the bacterial pathogen reproduces slowly. This is correct. Therefore, it takes a long time for the antibiotic to kill all the bacteria that's there. 3, the pathogen is not very sensitive to antibiotics. So this is also correct. Due to the massive antibiotic resistance, it also takes a really long time to kill all of the bacteria. Therefore, the answer is going to be D. Question number 39. The average sizes of some pathogens are shown. One type of air filter has been shown to be effective at preventing any pathogens of 1 micrometer or larger from entering the air system of a room. Based on their size and mode of transmission, which diseases would the air filter prevent from entering the air system of a room? So the air filter prevents pathogens of 1 micrometer or larger from entering the air system, which is 1000 nanometers. So let's look for the pathogens which are greater than 1000 nanometers. Is going to be Mycobacterium, Plasmodium, and Vibrio cholerae. However, due to the mode of transmission, only Mycobacterium tuberculosis would be prevented from entering the air system of a room. The reason for this is because this is the only airborne disease. Therefore, tuberculosis is the only correct answer, because Plasmodium and Vibrio cholerae are not airborne diseases. Plasmodium are transmitted by female anopheles, and Vibrio cholerae is a waterborne disease. Therefore, a filter would not prevent their spread. Therefore, the answer is going to be D. Question number 40. A successful vaccination program provides a level of immunity where the majority of a population is protected. There are several factors that can affect the success of a vaccination program. Which correctly shows the factors that can affect the success of a vaccination program? So, frequent mutations of a pathogen. So, if a pathogen mutates frequently, this means that we require a lot of booster vaccines. The reason for this is that vaccines are only specific for one antigen. So if a pathogen keeps mutated and the antigen keeps changing, this means that we require a lot of booster vaccines, which means that this would reduce the success of a vaccination program. Therefore, this is correct. Now, vaccination from 8 years old, age doesn't really matter in a success of a vaccination. Therefore, this is incorrect. Now, pathogen is able to invade T cells. Now, as we all know, T cells are required for immune activation, which is how the vaccine actually works. Therefore, if a pathogen invades the T cells, this means that the immune system would be weakened or suppressed. This means that the vaccination program is less likely to be successful, since a proper immune system reaction cannot take place. Therefore, active immunity cannot take place, therefore this would reduce the success, therefore this is the factor. Now, booster vaccinations needed frequently, this means that the vaccination program will take way longer, therefore this is also likely to reduce its success, therefore the answer is going to be B. Thank you very much for watching this video. If you feel that you found it helpful, I would highly appreciate it if you would give this video a thumbs up and subscribe to this channel.